friends, once again in the class. Today we are going to study about the structure and functions of our very important moiety from our body, that is DNA and RNA, in the subject biochemistry, that is BP 203T. So, we already know something about DNA and RNA, and then what we should know about DNA and RNA and the functions. In this lecture we are going to discuss about it. So, first of all, nucleotides and nucleic acid and nucleotide. What is the difference between nucleic acid and nucleotides? That is, DNA and RNA are the nucleic acid, and when this DNA and RNA are incorporated with several more things, that is one phosphate moiety and a sugar moiety, then it becomes nucleotide. So, deoxyribonucleotide is DNA and ribonucleotide is RNA. Deoxyribonucleic acid is DNA and ribonucleic acid is RNA. What are these? What is the main role of DNA and RNA? So, these help in the transmission of genetic material, we all know that, and generation to generation it helps us to maintain our special characters which we got it from our parents, and our offsprings will get it from ourselves. It was first discovered by John Frederick Missner in the year 1869, and DNA is a chemical basis of heredity which is regarded as the reserve bank of genetic information. DNA is exclusively responsible for maintaining the identity of different species of the organism. So, genetic material carrying is the main function of DNA and RNA both, and the interrelationship of these classes, that is DNA, RNA and ultimately protein, is called as central dogma of molecular biology. Now, let us discuss, it is a very important topic and very important question for our examination, that what is central dogma of molecular biology or what is central dogma of life. So, here we have three things: DNA, RNA and protein. When DNA replicates itself, that means when DNA forms its own copies, then it will be called as replication. When DNA converts some of its DNA into RNA, that means when there is a conversion of DNA into RNA, the process will be called as transcription, and when the RNA will be converted into proteins, then that process will be translation. And this whole thing, that means replication, transcription and then translation, is called as central dogma of molecular biology or central dogma of life. Then, if we see the structure of nucleotides, we have studied that there are three things present in a nucleotide. That means there is a nitrogenous base that will be purines and pyrimidines. There will be pentose sugar, that is ribose and deoxyribose, ribose in case of RNA and deoxyribose will be in the case of DNA, and a phosphate group. So, in purine we have adenine, guanine, and in pyrimidine we have cytosine, thymine and uracil. With a case, that is thymine is with DNA and uracil with RNA exclusively, that should be noted down. Pentose sugar, ribose in case of RNA and deoxyribose is in case of DNA, and the phosphate group which is common. So, here is the assembly of all the three things present here. This is the phosphate group, this is the sugar moiety, and this is your purine and pyrimidine bases. So, likewise they are getting connected through one another. That means, this is full one unit of nucleotide, this is another unit, this is another unit and this is another unit. And you can see this nitrogenous base is cytosine, this is guanine, this is adenosine and this is thymine. So, this is by knowing that there are four nitrogenous bases, that is cytosine, guanine, adenine and thymine, we can simply say that this is in the formation of DNA. Why I told you this? Because there is the nitrogenous base thymine present. If there is a nitrogenous base uracil, will be there, when the uracil will be there, that means there will be G, C, A and U. If this is the sequence or this is the nitrogenous bases present, then there will be, we are talking about the structure of RNA. So, in this structure we are talking about DNA. Next, nucleic acid we have seen in the previous lecture. So, this is the phosphate moiety, this is the purine or pyrimidine base and this is the sugar. What we are talking about here? Here is the presence of OH. If there will be a presence of H, then we will talk about DNA. Because there is a presence of OH, then we are talking about RNA here. So, I think is it clear? It is clear that by seeing the nitrogenous base using here and the sugar moiety, we can clearly say that we are synthesizing RNA or DNA. Clear? Next, DNA and RNA, they contains purines, adenine, guanine and pyrimidine, cytosine, and DNA has thymine in it, RNA has uracil in it. These are the structures of all your compounds present. That is, this is adenine, this is guanine, this is cytosine, this is thymine and this is uracil. These two are purines, whereas these three are pyrimidines. Next, tautomerism, it is playing a very important role in purines and pyrimidines. There are existence of a molecules in two forms, that is keto, we also call it lactam, and enol form, that is lactam. So, these two, when there is a presence of keto form lactam and enol form lactam, then we call it as tautomerism. The heterocyclic rings of purines and pyrimidines with oxo, that is C double bond O functional groups, that exhibit tautomerism. The purine, that is guanine and pyrimidine, cytosine, thymine and uracil exhibit tautomerism. So, it should be noted that in purines, guanine and in pyrimidine, cytosine, thymine and uracil, all of it will exhibit tautomerism. Now, nucleotide, they perform a wide variety of function in the body. That means they give us the building blocks or monomeric units for the structural components of, they are the structural components of DNA and RNA. At the same time, they also synthesize some of our coenzymes of beta complex, B complex vitamins, that is FAD and NAD positive, and also they help in the production of ATP, which is our energy molecule, which is our main source of energy. So, nucleotide gives more than 2 to 3 functions in our body. Some bases are there found in nucleic acids, those are very important for us, like 5-methylcysteine, cytosine, it is a derivative of cytosine, 5-methylcytosine. Then we have N4-acetylcytosine, again a derivative of cytosine. We have N6-methyladenine, it is a derivative of adenine. We have N6-N10 dimethyladenine, and we have pseudouracil. More we already have these very important compounds with us, and we also have some more compounds which are equally important. Then there are other biological important bases which are hypoxanthine, xanthine and uric acid, which are present in the free state. These are also the types, the parts of our nucleotides. Then now we need to understand what is the difference between nucleoside and nucleotides. In simple forms, I would like to say nucleosides have 2 things in it. Out of 3 things as in one is your pentose sugar, one is your phosphate group, and one is your purine and pyrimidine bases, that is nitrogenous bases. Nucleosides has 2 things in it, that is they have nitrogenous bases, they have the sugar moiety with them, whereas nucleotide has all the 3 things with them, that is they have the sugar moiety, they have the phosphate group and they have the nitrogenous base. So, how nucleoside and nucleotides are different? Nucleoside do not have phosphate group in them. Nucleotide have phosphate group in them. So, if anybody asks us what is the difference between nucleosides and nucleotide, we can easily say that nucleosides have two things in them. First is nitrogenous bases and the sugar moiety, whereas nucleotides have nitrogenous bases, sugar moiety and a phosphate group. That is why their name is, like we are talking about adenine, we are talking about adenine. So, in nucleoside it will be adenosine because there is no phosphate unit, and in nucleotide it will be adenosine 5 monophosphate or adenosine monophosphate because there is a presence of phosphate group. So, if anybody asks us, okay, what is the difference between nucleosides and nucleotide, we can easily say that nucleosides have two things in it out of three things, that is nitrogenous bases and sugar, whereas nucleotides has all the three things in it, that is nitrogenous bases, sugar and phosphate group. It is common with everything like RNA and DNA. We can simply say, we can simply define everything in it. Now, understand that if we are talking about RNA and DNA, like RNA also has adenine in it, DNA has adenine in it. How you are going to understand which adenine we are using for RNA and which is for DNA? So, we need to write the names likewise. Like for DNA, adenine will be called as adenosine in nucleosides, whereas in nucleotide it will be called as adenosine monophosphate. But when we are talking about DNA, how it will be called? Adenine will be called as deoxyadenine. Make sure, because we are here, we are using here deoxyribose sugar. Here in the case of RNA, we are using ribose sugar. That is why like this we can make a difference. So, what will be the name of nucleotide if we write it down for DNA? It will be deoxyadenosine monophosphate. That means if we write down for RNA, it will be adenosine monophosphate. If we will write down for DNA, it will be deoxyadenosine monophosphate. Okay. So, it will be common for three things: adenine, guanine, cytosine. We will do this type of changes in adenine, guanine and cytosine. What about uracil and thymine? We do not make any we are writing here AMP, and in case of DNA we are writing DAMP. Okay. So, in uracil we need not to write because uracil, there is uracil in presence of RNA, whereas thymine is in presence of is present in DNA. So, these two things are different in RNA and DNA, rest three things are same, that is adenine, guanine and cytosine. So, we need to make the changes likewise. Okay. Next, how these are binded together, that means how all the things like sugar moiety, phosphate group and a nitrogenous bases are binding towards binding with each other. The bonds which they are using, first of all the pentose sugar which is bonded with nitrogenous bases are with glycosidic bond. We have studied about this glycosidic bond in the carbohydrate metabolism, because these bonds, glycosidic bonds, are present in carbohydrates. They are used to bind two different carbohydrate units. So, here when your sugar is bounded with nitrogenous bases, we are using glycosidic bond. Then when your nitrogenous bases is bonded with pentose sugar, there will be a covalent bond, which is very important, very tough bond in the purine nucleotide. In case of pyrimidine, the glycosidic linkage is between N1 of the pyrimidine and C1 of the pentose. You can see this by the structures present here. This is the structure of adenine and ribose, the structure of AMP, and this is the structure of TMP, that is thymine and ribose sugar. So, how they are bonded, that means adenine is purine and thymine is pyrimidine. So, how these bonds are made here, it is written, it is drawn in this case. That means, when your ribose sugar is reacted is combined with your nitrogenous bases, that means C1 with N9, there will be glycosidic linkage. Then C1 is reacted with N1, it is, we are talking about DNA here, we are talking about there will be that can be DNA also, that can be RNA also, because AMP is common in both. No, specifically we are talking about RNA here because there is a presence of OH group. So, here we are talking about RNA, whereas here we are talking about DNA. So, we need to see both the things, that either we are joining which nitrogenous group is joining here, and the second thing which sugar moiety is here. So, by that we can say whether we are talking about RNA and or DNA. Okay. Now, purine, pyrimidine and nucleotide analogous. We have already studied many of them in our previous slides, in our previous lectures, but here I would like to tell you more about it. Like allopurinol, which we used in the treatment of gout and hyperuricemia, has the same structure analogous of our nitrogenous bases. Then 5-fluorouracil, there is a presence of uracil, 6 mercaptopurine, 8 azaguanine, 3 deoxyuridine, 5 or 6 azauridine, 5 or 6 azacitidine, 5 iodouracil, there are all are analogous of purine, pyrimidine, or you can say that nitrogenous bases. Okay. And so many examples are there. Then let us talk about structure of DNA. DNA has nitrogenous bases, DNA has sugar, that is deoxyribose sugar, and a phosphate group. So, and how we are writing DNA, that is deoxy. We use the word deoxy before writing the full thing, that is DAMP, DCMP, DGMP likewise. So, we use the word deoxy. If we see the structure here, this is you can say a piece of single strand of DNA. There are 2 strands, helical structure of DNA is there. So, from one strand a single piece we are using here, because we are here to see this is the nitrogenous bases, this is the sugar moiety and this is the phosphate group, and this sugar moiety is connected with the phosphate group of second joint, the whole thing. Then guanine, again sugar moiety. Sugar moiety is again connected with the phosphate group, and the sugar moiety phosphate group, phosphate moiety is joined with the sugar group, which is ultimately joined with the thymine, that is the nitrogenous bases to the other side. From the left side, the phosphate group is joined through your sugar moiety, and the sugar moiety at the right hand side joined through your nitrogenous bases. Okay. Likewise. And now let us talk about our Chargaff's rule. What is Chargaff's rule? Chargaff rule is very important and was asked many times in the examination. That there was a scientist called as Irwin Chargaff in late 1940s. He quantitatively analyzed the DNA, and he told that what his observation was, what his opinion was, that there is a molar equivalence between purine and pyrimidines, which is like you can say purines and pyrimidines. Purines are adenine, guanine, whereas pyrimidines are cytosine and thymine in case of DNA. Now, adenine is having equivalent amount as thymine, whereas guanine has equivalent amount of cytosine. That means, the amount of adenine is equal to thymine, whereas guanine is equal to cytosine. Here I am not talking about the bonding, the type of bonding, later on I will talk on. Here I am talking about the amount of it. This is equal to and not the bond in between present. So, the amount of adenine is equivalent to the amount of thymine and the amount of guanine is equivalent to the amount of cytosine. This was observed by Chargaff and it is very important milestone achieved when we were studying about DNA structure. So, now the next thing, DNA structure, this Chargaff rule applicable on DNA only. It is not applicable on RNA, because when we are making double or helical structure of DNA, there are two strands present. That means one has adenine, the other one has thymine, one has cytosine, the other one has guanine. So, there you can see there is equal amount of nitrogenous bases present. But what happens in RNA? RNA has a single strand. How you will say that adenine should be equivalent to thymine? Because there is no other strand, there is no double stranded. RNA is single stranded. So, RNA does not follow Chargaff's rule, only DNA follows. Okay. Then DNA is double helical structure, who it was given by Watson and Crick. We studied many times Watson and Crick model of DNA helix. It was given in the year 1953. They got Nobel Prize for this in the 1962, and the structure of DNA, that is double helical. You can see the structures. It is right hand helix. Right, if you see the telephone wires in your houses, that how they are rotated, how they are helically beautifully modified, that it is a right hand helix. DNA has a right hand helix, that means the both the strands, they are helical in the right hand direction. Okay. So, these are some characteristics which are very important. We have discussed some of it, and now the other one we are going to discuss. DNA is right handed double helix structure. The second thing, both the strands of DNA are anti-parallel, they do not parallel to both, like they are not like this, no they are not, and the width of your double helical structure is 20 angstrom, also called as 20 nanometer. The one turn is 34 angstrom or 3.4 nanometer, and they placed at a distance of 3.4 angstrom. Each strand is hydrophilic deoxyribose phosphate backbone. Hydrophilic means water loving. So, they has a hydrophilic deoxyribose phosphate backbone, and they are non-identical but complementary to each other. That means, if one strand has thymine in it at the first position, the second strand will have a compound which is complementary to thymine, that is A, that is adenine. If it has first position, it has thymine in first position, it will have adenine. They are not identical, but they are complementary to each other. That is why they are making equivalent structure. AT, that means adenine is double bonded with thymine, whereas guanine is triple bonded with cytosine. We will see the structure later on. See, you can see the structure here. This is adenine, this is thymine, there is a presence of double bond here, one and second, whereas it has guanine, it has cytosine, there is a presence of triple bond here. So, hydrogen bonds are formed between the purines and pyrimidines only, AT, TA, GC and CG. You will never see A is bonded with G or T is bonded with C, that is for sure. A will be bonded with T and G will be bonded with C and vice versa. Okay. The complementary base pair in DNA helix proves Chargaff's rule, we already studied about it. The template, there are two strands in DNA. First is called as template strand or sense strand, whereas the second will be called as anti-sense strand. That means the DNA has two strands in which only one strand will be used for the replication process. The other strand will not be used at the same time. Okay. So, one is template strand, only by using that strand we can make the new strands. These are the DNA conformations, that means there are three types of DNA, A DNA, B DNA and Z DNA. bDNA is the DNA which was searched and founded by Watson and Crick. These are the structures of bDNA, aDNA and zDNA. Okay. This is the denaturation, simple, that is an organic thing, DNA is an organic thing. So, there is denaturation possible if we separate the polynucleotide strand by changing the pH of the reaction or by increasing the temperature, there will be denaturation of the DNA. Now, let us come about ribonucleic acid. Ribonucleic acid, I have already told you that is a single stranded structure. DNA is double helical, RNA is single stranded structure. It will have all the three things in it, that means it has a phosphate group, it will be having a nitrogenous base and it will be having a sugar moiety in it. Chargaff's rule will not be followed by RNA. Alkali hydrolysis can denature RNA because again it is an organic thing, it will be denatured if we do some sort of reactions to it, like if we change the temperature, if we change the pH, if we do the alkali hydrolysis, there will be hydrolysis of RNA and it will give orsinol color reaction. It is very important that the RNA can be histologically identified. Previously they were identified by treating the RNAs with orsinol. And these are the types and functions of RNA. We have several types of RNA in which there are 3 RNAs which we importantly use when we have to discuss our central dogma of molecular biology, that is mRNA, tRNA and rRNA, that is messenger RNA, transfer RNA and rRNA. Rest of the RNAs are important, but they are not very important. They are important definitely, but these 3 RNAs are super important for us. These are the types. We know messenger RNA is 5 to 10%, transfer RNA is 10 to 20, written is there, it is 10 to 20% and ribosomal RNA is 50 to 80%. mRNA, how it helps? mRNA is synthesized in the nucleus as heterogeneous nuclear RNA, also called as hnRNA, and on processing, the functional mRNA enters the cytoplasm and participate in protein synthesis. And ribosomal RNA, as the name suggests, it has 2 subunit ribosome, that is 2 subunits are 60S and 40S subunits. Later on we will discuss it when we are going to discuss about our transcription process. And these are the tRNA, it is a very plays a very important role. This is the structure of tRNA, it has several arms in it. It has like an acceptor arm, it has an anticodon arm, it has a D arm, it has a T psi arm, it has a variable arm, and there are 2 classes of it, class 1 and class 2. So, through the structure we can see there are certain things tRNA can do. This was all about this chapter. Thank you so much. Happy learning.